You gotta give them
Can you,
the microphone.
Share the microphone.
yeah, can you give me some minute warnings so that I know?
2 minutes.
Okay. So thanks so much for having me. This has already been a really fascinating evening. So I'm going to talk to you about what I call engineering biology at social scales. How many of you feel like you have a really good working understanding of biotechnology? Great. Okay, I pitched this at the right level. All right, so we're going to talk about biology as technology. So I am really excited by biotechnology. I'm trained as a bioengineer originally. And I think that biology, and many other people here are also really excited about thinking about what biological technologies look like. And it ends up that biology is a powerful and valuable technology. It can make a number of things, from pharmaceuticals to better plants, to better ways to store data, to new ways to produce flavors and fragrances and tissues. There's a wide variety of things that biology can be used for. And one of the reasons that biological engineers like me are excited is because we want to program biological systems with the same level of ease that we program other types of systems. And we have new tools. So not only do we use traditional engineering principles, but we can think about how can you use the principles of evolution and reproduction in your biological systems design. And things are changing quickly in biotech right now. These little microbes, right, they can make a wide diversity of things, pretty much any molecular structure you can think about, and that's encoded in their blueprint, in their DNA. And we're getting better at being able to work with these systems through a variety of ways, in being able to read that blueprint, able to write that genetic blueprint, and then also being able to edit and evolve these systems. And it's interesting because we can work with them not only in these new ways, but also differently across space and time. So when you can think about a genetic code and a physical biological system, we're starting to make new transactions between a digital biological space, the A, C, D, T's, and G's of your genetic code, and a physical space of physical genetic information, physical systems. We're getting better at making even more complex biological systems. In the 1970s here, we first learned how to cut and paste DNA and make new valuable things. So in this case, on the left-hand side is Genentech in the 1970s, where many of the ideas around this was you take a little bit of DNA, put it in another system, and make something valuable, in this case, insulin. Now we're making things at a much more complex scale. Guy second to the left, I worked with him for a while up at UC Berkeley. He put 13 genes into a yeast to make artemisinic acid, an anti-malarial, and used that same system to help make fuel. Right? Then Craig Venter, you may see him just went over thinking about engineering whole genomes, small genomes, but still whole genomes. And then how do we put these into 3D construction? We're also reorganizing the way we work. So my PhD was basically in moving colorless liquids around. That's no longer the case for most people doing their PhD. They can get robots to do the majority of working with biological systems for them. This is that same company I mentioned with that artemisinic acid and the fuel. This is pictures from a company out in the East Bay that at this time, several years ago, was making 300,000 unique genetic strains of yeast a week and seeing what they would make. We can also operate differently across space and time. This is an example from Novartis, where instead of now seeing a new infectious disease, needing to ship it somewhere, take a sample, create a new vaccine and ship it back, they can run different types of experiments where they upload that genetic information, just put it over the internet, download it, make a new vaccine, upload it again, and then produce that new countermeasure locally. And in this case, it actually shifted the rate-limiting step in the development of a new vaccine from the technical production of the vaccine to the regulatory approval of the vaccine. So we're seeing these shifts in the way in which we can engineer biology really qualitatively change this practice. We're seeing reorganizations of industries, biotechnology, the bioeconomy, as we call it, is growing faster than the economy as a whole. And the people who are interested in working in biotech are really spreading all over the world. This is the iGEM competition. I'm going to speak about that a little bit more. And they really are all over the world. These new communities that want to be bioengineers, that really are training to be bioengineers and doing new fascinating things, are really diffusing all across the globe. It's important to remember, though, that in this case, the biology has already taken over the world, right? So we are now interacting with these living systems in new types of ways. And what I am really interested in now, working at this strange place called the Center for International Security and Cooperation on campus, where I've turned from a bioengineer into a policy wonk, is to figure out what this whole ecosystem of people and industries and technology looks like today. And most importantly, who is going to decide what is okay and not okay to do with biotechnology? This is one of my favorite comics from XKCD that shows maps of online communities. Here I think about who is shaping what is sort of your practices, what's allowable, what's okay on the internet. Who are going to be the Facebooks of biotech? And this is not an abstract thing. There are real policy challenges that are coming up with biotechnology that I won't have time to talk about today, but these are just some images from the headlines you see, right? New ways to drive genetic traits through populations, eliminate whole species, make designer babies, brew your own drugs at home, or grow your own in a Kickstarter kit. And so what is going to be the types of activities that shape whether or not we see this technology as really a weapon of mass construction, right? All this constructive, productive force, or a weapon of mass destruction, as the Director of National Intelligence proclaimed it to be a contributing force to in last year's threat assessment. Or what it is probably right now, which is a weapon of massive uncertainty and confusion about just what the heck is going on and who's in charge. So the questions that have been driving my research have been really around these microbes. How can we assure ourselves that getting better at engineering biology is good for the world? How do we know whether our governance systems, the rules, informal and formal, that we create, are working? And how can we engineer biology? How are we engineering biology? I mean, designing, building, and testing these social systems above the technology and underneath the technology at these social scales. So I'm going to talk really just about one venue in which we're trying to do this, and this is the iGEM competition. Who's heard of it? Few people? Okay, that's good. If you haven't, you can go with the sticker at the end. But first, before I talk about iGEM, I'm going to talk about FIRST. Who's heard of FIRST? Like a few more people. Okay. So when I was in high school, I did FIRST. FIRST is a robotics competition. Right? Basic idea, you get a kit of parts, you get something to put them in, and then you have to build a robot that plays a game. I'm Canadian, so our robot had to play hockey. Of course, we had grand ideas about what we were going to engineer. We designed that system, and we thought, our robot's probably going to look something like this. We got, a little more humble over the summer. Looked a little bit more like this. That's actually not true. It looked like this. It was like a stick on a robot. We got last place at this great competition against other robots. Despite this, it was one of the most exciting and formative things and taught me that I wanted to be an engineer. Many of the same people who are trained as engineers wanted to do this with biology, and they created iGEM, the International Genetically Engineered Machine Competition. Same idea, you get a kit of parts, but these are genetically encoded parts. And instead of a garbage can, you're putting it in a bacteria. This is what it actually looks like. It's really hard to see the parts, but the basic idea of them is they call them biobricks. They're standardized genetic architecture, standardized genetic parts that can be assembled in a standard way to produce complex functions. They started this competition when I started grad school at MIT in about 2003, 2004 as a winter course. And instead of getting the bugs to battle against each other, which is a really bad idea that we can talk about later, they asked, what would you do with biology? And in the first case they thought of, bugs smell bad, let's make them smell better. So they said, let's encode a genetic part that when these bacteria grow, they smell like bananas. And so they engineered a banana odor generating machine. And then they thought, let's make another one. Let's make a wintergreen odor generating machine. Put them into a bacteria together, and then depending on whether or not the bacteria were growing or in stasis, it would smell like bananas or like wintergreen. Didn't work the first time, but they had the idea. They put in their aggregated DNA part device system, and they described it. And they submitted it to an online toolbox, a registry of standardized biological parts. And then they presented their ideas to their peers. They wrote about it online, on their wiki, and they got a prize. And of course, it's a big brick. And everybody got really excited. And it was a place to share this new way, this new type of engineering in biological systems, right? Even if it was just in bananas. And this idea grew. So 2003, this is the group. This was last year. Last year we had about 5,000 students in 300 teams from 42 countries participating. They all descended on Boston in the winter. This is where they came from. They don't just make banana smells, they do everything from diagnostics to energy to environment to food and nutrition, advances, manufacturing, new applications. There's even new collaborations with art and design. You can have an art and design track. And some of those people have gone on to be incredibly successful. The guy on the right was on that banana generating team. His company, now in Boston, has raised hundreds of millions of dollars in venture capital in order to grow a company around making any molecule you want, any engineered organism you want, including a fragrance from an extinct rose. This type of generative competition structure allows us to do really interesting things looking at the incentives on what distributes norms and practices. And what is really interesting about the competition is we don't just give out prizes for technical expertise and technical excellence. We both require incentives in terms of fulfilling certain safety requirements, show us that you're following the rules and addressing any rules where there are ambiguities and gaps. And then we also give prizes for what we call human practices, which are innovative approaches to dealing with all those messy policy issues and practices issues that might show up in the context of your individual experiment. Then we have a committee that reviews all of these experiments every year, the hundreds of experiments every year. And then we can change the rules and see what happens. We can change the incentives, the training, the examples, the mentors that we give them. So 13 years, and I think we have 30,000 students, alumni that have participated in the competition. I'm just going to give you a couple little highlights of some of the things we've seen from this sort of human practices and safety side. This is one of my favorite examples. This is a team from London several years ago now, and what they wanted to do was they wanted to figure out how their biological systems were working, and they wanted a way to actually observe the output. And so they engineered pigments that would show up in their bacteria. So these are actually the pigments that they engineered, and they set it up to a system so that depending on the input, depending what was on the environment, the bacteria would turn different colors. And then because they had to figure out, like, what are all the issues that could come up when we engineer these systems, they partnered with actually some artists and designers and then went through a process of speculating on what are possible futures and uses of this, good and bad. This is one partnership that they came up with, And imagine, what if we use this in a drinkable probiotic that, depending on what's in your gut, you could get a readout of the good and bad condition of your gut. So they actually, the Scatalogue was shown as an exhibit now most recently at the MoMA on showing these new types of partnerships between technologists and artists. And we have hundreds of these examples. On the safety and security side, we've also done new types of partnerships with incentives and mentors, including some interesting partnerships with groups like the FBI. The FBI is now a major sponsor of the IGEM competition. This is an effort that I like to call Know Your Local WMD Coordinator. So every year, the local WMD coordinators get up on stage and talk about what we do and don't do with biotechnology, and how these students from all over the world can help to enforce the types of positive developments in biotechnologies and mitigate against those possible risks. They partner with teams. So this is a team from Lethbridge, Alberta, that worked with the FBI and local companies to triage the ordering systems and actually be white hat biohackers and see what are some potential vulnerabilities in this system. They presented on this and won a safety prize. And then the students flip the equation. Here are IGEM students presenting at the UN Biological Weapons Convention about the innovations that they have made in developing policy and practices that have exceeded, in many cases, these formal institutions. And these safety engagements have actually caused new communities to then participate in the competition. So this is our first team from Pakistan this year that won a bronze medal. And they're showing the judges and the mentors vulnerabilities that we may not have caught in the system. And they're teaching some of their heroes. So this is George Church on the left, who's one of the leading genetic engineers in the country, listening to a team, I think this is a team from Singapore, talk about their project. And they're starting to show up in the news about the types of innovations really exceeding everybody's expectations on what they can achieve. So how are we engineering biology at social scales? I've shown you a few anecdotes, but the really big question that drives me, I won't be able to answer today, but hopefully I'll come back at some point, is talking about how do we know at a more systematic level whether or not these rules are working? What types of perverse incentives am I giving to undergraduates all over the world every year? And so part of my research program here is actually looking at these questions in a much more systematic and rigorous way, not just looking at IGEM, but looking at a number of other venues, including this Sinberg organization and a large national lab working in biotechnology, looking at these variables and interventions, and then both looking at the teams that, what people think are problems in safety and security and policy, and then whether or not these square with expert assessments. So is there agreement, is there a robust agreement on what we think are good or bad practices, and how do these change over time, how do these change over geographies, and how do these change over other types of institutional factors? And working with some pretty awesome data scientists and others to begin looking through all of those archived wikis and what we can learn from them and the genetic repositories and begin to try and draw the connections between these over time and how these are referenced through the types of incentive structures, the medal criteria, the judges, and whatnot. And hopefully I'll be able to report back next year, and even though I don't have results yet, there'll be thousands more students next year. I wish I could tell you a lot more about some of the experiments that we're doing here on campus. If you're interested, I'd love to talk to you later. Thanks.